Swayam Prabha. Digital India. Educated India.
This is the third lecture in this particular course, and in this lecture we are going to talk about diversity in general transcription factors. Now, so far we have discussed about the diversity in RNA polymerases in eukaryotes. There are 1, 2 and 3 RNA polymerases, and more recently there is a RNA polymerase 4 which was discovered in plants. Then we discovered that there are what are called as core promoter elements to which these general transcription factors in RNA polymerase bind, and that is what contributes to transcription. And in the last class I have actually mentioned the core promoter elements means not just Tata box, there are many variants of these Tata sequences, and these variations in Tata sequences actually contribute to differential gene regulation. And I gave you a number of examples of like a initiator motif, BRE, DRE and so on and so forth, how variations in core promoter sequences actually contribute to differential gene regulation. And I also mentioned to you that these core promoter sequences are very important not only for transcription, but they also play a very important role in transcription activation by upstream activators. There are many transcription factors which bind to upstream sequences called enhancer sequences, and they function only when a specific core promoter element is present. For example, I gave you examples where even within the Tata sequence, if you replace a Tata sequence of one promoter with the Tata sequence of another promoter, transcription activation cannot take place by a transcription activator. So, variations within these core promoter elements themselves contribute to diversity in gene regulation. So, the job of core promoter elements is not just to contribute to initiation of transcription, but they also contribute to differential gene regulation. That was the crux of the previous two talks. Today we are going to focus on the general transcription factors are also diverse. Now, there is not just one TF2D or there is not just one TBP, there are variations of general transcription factors, and this diversity in general transcription factors can also contribute to differential gene regulation in eukaryote. That is what is going to be the crux of the talk. So, just to recapitulate what we discussed so far, we discussed so far about differential gene regulation by core promoter variants. This I discussed just now. There are many different core promoter elements in eukaryotes, not just the Tata box, there are many variants of these sequences, and there is nothing like a universal core promoter element in eukaryotes, because not a single core promoter element is present in all the Tata boxes. In all the eukaryotic promoter, some of them contain initiator sequence, some of them contain Tata motifs, some of them contain a BRE, DRE and so on and so forth. Core promoters are essential not only for the assembly of RNA polymerase machinery and transcription initiation, but they are also essential for the modulation of function of other upstream cis-acting elements known as enhancers, as well as transcription factor which bind to enhancer sequences. I just discussed with you, there are some enhancer sequences, they function as enhancers only when certain core promoter elements are present downstream. If you replace these core promoter elements with the variants, these upstream enhancers can no longer function. Therefore, there is a link between these different promoter sequences in a eukaryotic promoter. And what now we are going to focus today is that how diversity in general transcription factors interacting with the core promoter elements can also contribute to differential gene regulation. That is going to be the crux of today's talk. Now, this is again to give you a overall perspective, because I do not want it to be under impression that we are talking only about general transcription factors and there are other regions which are not important. If you know, just like I gave an example of a cricket pitch in the last class, when you talk about a cricket match, you have players, you have different players, you have close-in fielders, you have outdoor fielders, but all the action is focused only on the cricket pitch. So, the promoter is analogous to the cricket pitch, and you have different batsmen, different bowlers coming in and doing just in the same way. You have different protein factors which contribute to differential gene regulation, and you also have what is called a close-in field, outfield, and then you have a stadium and so on and so forth. In the same way, you have proximal promoter elements, you have distal promoter elements, you have enhancers, silencers, boundary insulate elements. All these ultimately contribute to overall regulation of the gene expression. But today we are only going to focus upon general transcription factors which are binding to the proximal promoter sequences, and how that alone can bring about several levels of gene regulation. That is going to be. So, in the subsequent classes we are going to talk about other promoter agents like enhancers, silencers, boundary elements and so on and so forth. Now, Transcription activation, so far we discussed only about cis acting elements. In the last class we only talked about the cis acting elements which are present in the core promoter region. Today we are going to talk about trans acting factor, that is the protein factor which act in trans, and what kind of transcription factors are actually involved in transcription activation in eukaryotes. Generally there are about broadly 3 classes of trans acting factors which play important role in transcription activation in eukaryotic promoters. The first set is called the general or basal transcription factors that interact with the core promoter elements, and today's talk is going to focus on this particular set of protein factors. There are also transcription activators which will bind proximal and distal promoter sequences. We will come to it at a later stage in this course. And there are also very importantly what are called as co-activators or co-repressors which act as a bridge between transcription activators and general transcription factors. So, just to give you a very brief overview, you have a set of sequences called core promoter sequences, you have a set of sequences called proximal and distal promoter sequences, enhancer, silencer and so on, which are present far upstream. And there are a bunch of protein factors called general transcription factors which bind to the core promoter sequences, and there are a bunch of protein factors called enhancer binding proteins or transcriptional activators which bind to far upstream sequences, and more importantly there are a whole bunch of protein called co-activators or co-repressors which actually acts as mediators between the upstream activators and the general transcription factors. So, they act as a bridge between these 2 sets of protein factors. So, today's talk will focus only about the general transcription factors which bind to the core promoter elements and how variations in the general transcription factors can also contribute to differential gene regulation. Now, so we mentioned in the first class very briefly how RNA polymerase in conjunction with 6 general transcription factors, namely TF2A, B, D, E, H and F, can actually bring about transcription initiation in eukaryotes. Right. Now, I also mentioned very briefly in the first class each one of these general transcription factors has 1 to many subunits, and they have different molecular weights and each of them actually have a different function. For example, the TBP component of the TF2D actually is involved in promoter recognition and its job is to actually also recruit the TF2B. And there are also the TF2D in addition to TBP also contains what are called as TBP associated factors, and they actually play a very important role in transcription activation and promoter recognition. You have TF2A, B, F and E, and each of them have a specific function during transcription which we discussed in the first class. Now, I also mentioned very clearly that enormous amount actually on time and effort has actually been spent in purifying each one of these general transcription factors. For example, people actually have taken nuclear extracts devoid of histones and DNA and then fractionated them on ion exchange columns and then eluted proteins by using different salt concentrations and then rechromatographed in different kind of ion exchange columns, and based on all this differential chromatography of various nuclear extract fractions, proteins, each one of the general transcription are purified in different set of these column chromatography fractions, and each one of them are purified, and then all these purified transcription factors were actually put back, and then when you put a promoter sequence containing a downstream coding region and you put nucleotides, you have been able to demonstrate that transcription initiation can actually be mimicked in a test tube. And this is what is called as a cell-free transcription, and these cell-free transcription assays actually played a very important role in the analysis of promoter sequences as well as in identifying what are all the various components of the general transcription factors. So, today let us now start discussing about TF2D, how diversity in TF2D can contribute to gene regulation. That is what we are going to begin with in this class. Now, just to recapitulate about TF2D, TF2D is a multi-protein complex, and the protein responsible for recognizing the Tata box in the TF2D complex is the Tata binding protein or the TBP, which we all are aware. There are many other proteins, about 10 to 14 different proteins of the TF2D complex, and these are actually referred to as the TBP associated factors or TAFs. Remember this word TAFs, TBP associated factors. We are going to discuss a lot more about this particular TAFs in this class. Now, TBP can specifically bind to Tata box on its own. So, although TF2D consists of TBP and many other TBP associated factors, if you just clone the gene coding for Tata binding protein, express the recombinant recombinant Tata binding protein, and if you now take a Tata box sequence, the Tata binding recombinant protein can actually bind to Tata box very well. And then you ask the question, why do you require TF2D? What is the function of all these TBP associated factors? This was actually demonstrated when they actually did cell-free transcription using this recombinant or the purified TBP alone, as well as TF2D purified from cell extracts which contains TBP as well as TAFs, they found a very important difference. Now, TBP although can support basal transcription, TBP could not support activator dependent transcription. That means, if you have a promoter region which contains not only the core promoter sequence, but it also has the binding site for a transcription factor, for example in SP1, which is a specific transcription factor which binds to a proximal or distal promoter element. When you take such a promoter sequence which contains a core promoter sequence as well as the binding sequence for an upstream activator, and if you now put only TBP instead of TF2D, you see TBP, the level of transcription is more or less same. Addition of TBP does not make any difference. So, the SP1 cannot activate transcription when you have only TBP, whereas when you have TF2D instead of TBP, then you see SP1 is able to activate transcription, which clearly these kinds of experiments clearly told that the TAFs of the TF2D actually are essential for mediating the activator function of many upstream activators. TBP cannot substitute the TAF functions, and TBP in combination with TAFs are essential for activator dependent transcription in cell free transcription assays. So, this was the first demonstration that the TAFs of the TF2D complex play a very important role in supporting activator dependent transcription in eukaryotic cells. So, studies in the late 1980s revealed that in addition to TBP, certain TAFs also bind to core promoter elements, suggesting that in addition to the diversity in core promoter elements, diversity among proteins binding to core promoter elements may also contribute to differential gene regulation. So, this is the first point I want to emphasize in this particular class. The last class I told you diversity in the core promoter elements itself contribute to differential gene regulation, but now I am going to tell you diversity in the TAFs and diversity in TBPs can also contribute to differential gene regulation. This was discovered sometime in the late 1980s. The components of TF2D including TBP and TAFs have many paralogs. Now, what are paralogs? Paralogs nothing but they are related proteins encoded by different genes belonging to the same family. So, when I, for example, when I identify TBP in humans and if I identify a TBP related protein in Drosophila, that becomes a paralog. So, they all belong to the same TBP gene family, but there is a paralog of TBP which is a TBP related protein which I have identified in Drosophila. So, many paralogs of TBP and TAFs were discovered in many eukaryotic species. So, as people started looking at general transcription factors in Drosophila, humans, insect cells and yeast and so on and so forth, they started identifying that there are many members of these gene families. There are many variants of TBP, there are many variants of TAFs, and these are actually called as paralogs. And what was very interesting, when people started identifying some of these paralogs of the general transcription factors, many of these paralogs or many of these variants of general transcription factors have been found to be expressed in a tissue and development specific manner. This was the first indication that the general transcription factors themselves are differentially expressed in certain cell types, and therefore, they can themselves contribute to differential gene regulation. So, just as I told you in the last class, core promoter elements job is not just to support transcription, variations in the core promoter sequences can also contribute to differential gene regulation. Today I am going to tell you the job of the general transcription factors like TF2D is not just to assist RNA polymerase to come and bind to the core promoter region and initiate transcription, but there are variations within these general transcription factors, and these variations also can contribute to differential gene regulation. For example, there are variants of TBP, and these are actually referred to as TBT related factors or TRFs. So, variants of TBP have been identified, and they seems to be, for example, certain TBP related TRFs or TBP related proteins bind to only certain promoter sequences, but not the others. Similarly, there are certain TAFs, they themselves can actually bind to certain core promoter sequences in addition to TBP. So, all these variations actually contributed to differential gene regulation. So, the excitement in this area of eukaryotic gene expression, a lot of action took place in the early 70s to 2000. That is where there was actually a burst of activity in the eukaryotic gene regulation. A number of research groups worked on, dissected out various facets of both activator dependent transcription as well as general transcription factors in model systems like Drosophila, yeast and mammalian cell extracts. And because of these studies, a new knowledge actually started flowing in the late 1980s after 2000, and a number of very interesting observations actually came out of these research activities. So, the excitement actually began with the observation that certain components of TF2D are cell type specific. Now, so far you may have been under the impression that general transcription factors, because they are actually present are required for transcriptionation of all protein coding genes, they must be expressed in all cell types. So, whether you make TF2D from HeLa cells or Drosophila or yeast cells, they should all be the same, right, that must be the assumption. But soon people realize that when you make some of this TF2D from certain cell types, they are not the same, they are different, suggesting that there are many variants of the general transcription factors which are expressed in a cell type and tissue type specific manner. So, the so called basal apparatus comprising of general transcription factors and RNA polymerase II that forms the heart of the pre-initiation complex may actually exist in different cell type specific forms. So, the first important observation that I want to discuss with you is that there are many general transcription factors which are expressed in a cell and developmental specific manner. So, they are not the same as we find in the normal cells. For example, in the case of TAFs, once they identified a bunch of TAFs from model organisms like Drosophila and yeast cells and HeLa cells and so on and so forth, they identified a new TAF called TF4B, which is actually expressed only in the ovarian cells in humans. And this kind of a variant of TF4B is not exactly the same of the TAF4 which is expressed in other cells, suggesting that there are actually variants of these TAFs which are expressed in other cell types. So, once they found these kinds of things, people started looking at TAFs in other cell types and other tissues, and such variants of TAFs were identified in number of other organisms like flies, mice and humans and so on and so forth. Similarly, the Tata binding protein is not just one protein. People also realized that when you started purifying TBP from other cell types and other tissues in other organisms, variants of Tata binding proteins called TBP related factors were identified, and they seem to be having functionally slightly different from the regular Tata binding protein. So, all these studies indicated one thing. Just as in the first class I told you that sigma factor switching in prokaryotes, that means by using different sigma factors capable of binding to slightly variant -10 -30 sequences, differential gene regulation is brought about in the prokaryotic cells. The same way, differential gene regulation in eukaryotes can be brought about by diversity in general transcription factors which are expressed in a cell, tissue and developmental specific manner in eukaryotes. So, this is the first point I wanted to remember in this class. Just as diversity in sigma factors contribute to differential gene regulation in prokaryotes, diversity in general transcription factors such as TBP and TAFs can contribute to differential gene regulation in eukaryotic cells. That is the first point you have to remember. Now, let us now try to focus on what kind of variations in TAFs contribute to differential gene regulation. Remember, what are TAFs? TAFs are nothing but TBP associated protein factors which are actually is a form of the TF2D. So, TAFs in association TBP constitute the TF2D, and many of these TAFs, the important function of TAFs is that they act as a bridge between upstream activators and the general transcription RNA polymerase. For many of the transcription factors like SP1, to activate transcription you require TAFs. If you provide only TBP, these upstream activator proteins cannot activate transcription. But the moment you provide TF2D which contains TBP plus TAFs, these upstream activators are able to activate transcription. So, people thought the only job of TAFs is to assist many of these upstream activators to activate transcription in a better manner. But the first evidence for the presence of a tissue specific TAF came from the identification of a TAF4 variant called TAF4B by Dickstein et al. In 1996 in cultured B cells. Now, TAF4B was later found to be present in mouse testis and ovary as well, and when you knock out this TAF4B gene in the females, they became infertile due to deficient folliculogenesis, and males exhibited age dependent infertility as a result of defect in this spermatogenic maintenance. So, this is very interesting. I wanted to pay attention to this. For the first time, people have realized that there are certain TAFs which are expressed in a cell and tissue specific manner. For example, there is a particular TAF called TAF4B which is expressed in ovarian tissues and the testis, and if you knock out this particular gene in mice, then certain gonadal functions are affected, very clearly indicating that this particular TAF may be important in a set of genes which are involved in spermatogenesis and oogenesis. So, you can see this was never foreseen before. People thought that all the TAFs are the same and their job is to assist form a TF2D complex and assist general transcription activator proteins to bind and activate transcription. But once they started identifying cell and tissue specific TAFs, and these TAFs seem to be involved in regulation of a specific set of genes. So, specific regulation of specific genes seems to be brought about by the expression of specific TAFs in specific tissues. So, TAF4B was shown to be required for the transcription of a subset of ovarian granulosa specific genes as well as genes required for spermatogonial stem cell maintenance. So, you can see these particular TAFs, which are expressed only in certain gonadal tissues, seems to be involved in the regulation of a subset of genes in these gonadal tissues. So, they are not general, they do not have a general function, they are not involved in general activation of general initiation of transcription of all the gene, they seems to be involved in the activation of a specific subset of genes. So, TAF4B was identified as a key modulator of extracellular signals that specify developmental and proliferative program of granulosa cells in the ovary. So, for the first time a tissue specific TAF was identified which seems to be having a very specific tissue specific function and seems to be involved in the regulation of a subset of genes in these particular tissues. So, such TAFs were now identified in a number of other tissues and a number of other organisms, and the purpose of this talk is not to tell you everything or discuss about all the TAFs, but just to give you one or two examples just to drive home the point that variants of TAFs can actually contribute to differential gene regulation. That is the crux of today's talk. So, the importance of tissue specific TAFs in gene regulation was further strengthened by the identification of 5 tissue restricted TAFs, what are called as T-TAFs. T stands for testis. Testis specific TAFs are a testis restricted TAF subunits in Drosophila in the year 1996. And these 5 testis restricted TAFs are named, one of them is for example named as no hitter, which is actually a paralog of TAF4, cannonball, which is actually a paralog of TAF5, meiosis 1 arrest, which is involved in the paralog of it's a paralog of TAF6, spermatocyte arrest, which is a paralog of TAF8, and is called Ryan express, which is a paralog of TAF12. So, variants of these TAFs were actually identified and all of them seems to be expressed in testis, indicating that these paralogs of TAFs actually have a specific function in the testis. So, in the year 2001, Hiller et al. Actually demonstrated that disruption of each of these T-TAFs result in alteration of expression of tissue specific testis specific genes, and together these TAFs actually form a stable complex required for meiotic cell cycle progression and normal spermatid differentiation. So, you can already see how the TAFs which were actually discovered as general transcription factors required for transcriptionization of all the eukaryotic protein coding genes, it soon became clear that there are certain TAFs which are actually expressed only in certain tissues or certain cell types, and they also are involved in regulation of only certain specific genes involved in a specific physiological process. Similarly, in the case of mice, for example, a variant of TAF7 called TAF7I was shown to be expressed in testes in late spermatocytes and haploid spermatids. So, TAF5 knockout mice again exhibit altered patterns of spermatocyte gene expression and defects in spermiogenesis resulting in reduced fertility. So, you can see here are certain TAFs which are actually essential for spermatogenesis, and if you block the expression of this particular TAF, it results in infertility or reduced fertility in these mice. So, several such TAF variants have been identified in recent times, indicating that diversity in TAFs can actually contribute to differential gene expression in eukaryotes. So, the presence of tissue specific TAF variants enables the TF2D to work in conjunction with germ cell specific factors, cofactors or other components of the general transcription machinery and contributes to differential gene regulation. So, you can see again we have to come back to the example of cricket pitch, where for example there is a specific cricket pitch which is suitable, let us say, for a spin bowling. In the same way, there are certain promoters in certain tissues like testes, and in these promoters are actually present in say subset of genes, and these promoters are actually bound by specific kind of TAFs, and therefore these TAFs function in conjunction with specific upstream activators which are actually expressed in only that particular tissue, and in combination they actually are involved in regulation of a specific subset of genes. So, they contribute to differential gene regulation. The other important observation that came about in the case of TAFs is the discovery that TAFs actually contain very important enzymatic activities. In fact, many of the TAFs are actually identified as enzymes. This is another important discovery in the area of eukaryotic gene regulation, which actually changed the entire paradigm of eukaryotic gene regulation. So far we discussed TAFs as only proteins. No, their job is to form multi subunit complexes and assist upstream activators to activate transcription and help to form a TF2D complex. But the moment they realize that some of the TAFs actually have enzyme activities that actually change the way the eukaryotic gene expression was actually studied. Let us now understand what exactly we mean by this. Many of the TAFs actually possess multiple enzyme activities to post-translationally modify histones and transcription factors, thus allowing TF2D to serve as a core promoter binding factor in the context of chromatin and as an activator mediating activator response. Now, so far we have been discussing as if the DNA is a naked template. Now, in eukaryotes we all know that the DNA is actually present as chromatin, where histones are actually bound to the DNA template and the histones are organized in a very specific manner, contributing to chromatin formation and chromosomes. So, people soon realize that when you study transcription regulation, you have to explain how these transcription factors are regulating not just naked DNA templates, but you should actually explain how actually these transcription factors are regulating gene expression from chromatin templates. So, the first question that came to your mind is, if the DNA is covered by histones, how is this transcription factor is able to bind to DNA and activate transcription? The first thing that has to happen is you have to dislodge the histone. So, this is where when people actually realize that some of the transcription factors, especially TAFs, they actually can modify histones. For example, the human and Drosophila yeast TAF1, which is a TF2D specific subunit, it actually can acetylate histone. So, this actually can function as a histone acetyl transferase or HAT, which is capable of acetylating histone H3 and H4. The moment they discovered that some of the general transcription factors such as TAFs can actually add a phosphate or add an acetyl group to the histones, it became very clear that the mechanism by which many of these transcription factors are regulating gene expression of chromatin templates is actually by post translationally modifying histones. See, very simple. Now, histones are basic proteins, right? So, DNA is negatively charged. So, the basic protein which has a positive charge can bind very tightly to the DNA, right? But the moment these TAFs go and add acetyl group to the histones, now the histones start becoming negatively charged. And therefore, this negatively charged histone cannot bind very tightly to negatively charged DNA, therefore, the histones fall off and now these transcriptional activators can now bind, pre-initiation complex can be formed very clearly and therefore, transcription can take place. So, the discovery that TAFs actually contain specific enzyme activities changed the whole field of eukaryotic gene expression. People recognize that the assembly of pre-initiation complex and the binding of specific transcription factors is actually facilitated by selective removing of histones by some of these transcription factors by post-translationally modifying histones. Very simply, if you want to shut off the expression of a particular gene, you simply add a phosphate, you simply add a positive charge, you methylate histones. So, DNA histones become more positively charged, so they will go and bind DNA much more tightly, whereas if you want to activate transcription, you add a negative charge to the histone, therefore, histone fall off, now the pre-initiation complex can be formed very nicely and therefore, transcriptionation can take place. So, by simply making histones bind or fall off by either adding a positive charge or by adding a negative charge or by removing a positive charge or by removing a negative charge, by using what are called as histone acetyl transferases and histone deacetylases, histone phosphorylases and so on and so forth, differential gene regulation can be brought about, and one important factor player in this entire area are the TAFs. So, many of the TAFs soon discovered to be enzymes capable of post-translationally modifying histones. Similarly, for example, the TAF1 also has been able to act as a kinase by actually phosphorylating histones. So, many of the TAFs can act either as histone acetylases or they can actually act as kinases by either acetylating histones or phosphorylating histones. So, this discovery now explained very clearly why certain TAFs are actually required for activation by certain transcription factors. Now, this slide we actually discussed in the last slide, where I gave an example how TBP, purified TBP or recombinant TBP can only support basal transcription, but when the TBP cannot support activator dependent transcription. You can see here in the first lane you have only the promoter sequence, so you have only the basal transcription and the general transcription factors. When you add TBP and SP1, there is no change in the level of transcription here. Similarly, when you add SP1 to SP1 cannot actually change the basal transcription, but instead of TBP, if you now add TF2D, and you can see SP1 in conjunction with TF2D can enhance transcription. That means for SP1 to activate transcription, you cannot, TBP alone cannot do the job, you require TBP plus TAFs, which is actually the TF2D. So now we explain what the TAFs are actually doing. So in order for SP1 to activate transcription of chromatin templates, some component of the TAFs are actually required, and these TAFs are probably removing histones in the vicinity of the promoter, and therefore it can facilitate pre-initiation complex, and thereby it facilitate SP1 to activate transcription from these promoters. So the ability of some of the TAFs to modify histone to explain how only TF2D, which consists of TBP and TAFs, but not TBP alone, can enhance activity dependent transcription from chromatin templates. This is a very, very important observation that changed the entire field of eukaryotic gene regulation. So only TF2D rather than TBP can work in conjunction with TF2D deficient Pol II whole enzyme complex to facilitate activity dependent transcription from an in vitro reconstructed chromatin template, suggesting that a unique involvement of TAFs in the chromatin transcription. So the moment people started studying chromatin templates and asked the question how general transcription factors and activators activate transcription, the observation that chromatin dependent transcription actually involves modification of histones, and many of the transcription factors, especially the general transcription factors, actually contain histone modifying activities. This actually changed the entire field of eukaryotic gene regulation. So in addition to modifying histones, many of the TAFs such as TAF1 either can covalently modify other general transcription factors and cofactors as well. I will give you one example. TAF1 for example can deacetylate one component of TF2E. It can also phosphorylate an RNA polymerase associate protein called RAP74. It can also modify histone H2B, PC4 and the beta subunit of TF24, and also what is called as ubiquitination of TAF5. So what these examples tell you is that the TAFs or the TBP associate proteins actually are enzymes, many of them, not all, many of them are actually enzymes capable of covalent modification of number of other proteins, and these covalent modifications can be either acetylation, it can be phosphorylation, ubiquitination, so on and so forth, and all these things have a very important role in gene regulation. Okay. So far I told you TAFs, the TBP associated proteins actually can have enzymatic activities, and there are actually variants of TAFs in certain tissue types and certain cell types. Specific forms of TAFs are expressed, and these specific TAFs can actually bring about tissue specific and cell or developmental specific gene regulation. Now I am going to tell you, in addition to TAFs, there are variations on TBP itself. Now let us look at it. Now all multicellular organisms contains at least 2 genes coding for TBP family members that share sequence homology at their C-terminal 180 amino acid core DNA binding domains. As you know, TBP is a Tata box binding protein. That means it need to have what is called a DNA binding domain, and this DNA binding domain is actually present in the C terminus of the Tata binding protein, through which it actually binds and recognizes the Tata box. And usually the TBP, when you identify genes coding for TBP, this C terminal domain is actually conserved in many of them. The first gene which was identified as a one that coding for TBP, which is actually considered as the universal Tata binding transcription factor, is present in all the eukaryotes, and its job is to bind to Tata box. Now the second gene encoding a TBP related factor called TRF2, which is also known as TBP related protein TRP, TBP like factor TLF, or TBP like protein TLP. These are all various nomenclatures for the same protein, and this TRF2 actually shares about 60% sequence homology and 41% identity in the C-terminal domain of TBP, but very importantly, this TRF2 or the TBP related factor actually recognizes a sequence which is quite different from the Tata sequence. So you can see a variant of TBP itself can contribute to differential gene regulation because it recognizes a sequence that is quite different from the Tata box. Although it has sequence identity with TBP, functionally it actually recognizes a slightly different sequence than the Tata sequence. So this identification of TBP related factors again added a new paradigm to gene regulation, wherein variants of TBP themselves can activate different genes depending upon what kind of core promoter sequences they recognize. So in addition to this TRF2, 3 species specific TRFs were actually identified. These are called as TRF1, TRF3 and TRF4, and the TRF1 was actually first identified in Drosophila and was shown to be involved in the regulation of a subset of genes involved in nervous system. It is expressed only in nervous system of many of these genes, and is involved in the regulation of specific genes in the nervous system and gonads. The TRF1 actually binds TF2A, TF2B. It has its own set of TAFs which are called as N TAFs, which are specifically expressed in nervous tissue, and actually promotes the formation of pre-initiation complex in the nervous tissue. So, you can see very clearly there is a specific form of TBP which is called TRF1, which in association with TAFs which are expressed in specifically in neuronal tissue can regulate a subset of genes in the nervous tissue. So, tissue specific gene regulation is brought about by a variant of TBP and associated TAFs. For example, in Drosophila, a gene known as TUDOR has two promoters, one with a Tata box that binds TBP and another with a box called TC box that actually binds TRF1. So, TRF1, TBP both are belong to the Tata binding protein family, but the TBP actually recognizes the Tata sequence, whereas the TBP related factor actually recognizes a different sequence called as the TC box, which is present. This is present about -33 to -32 -22, whereas this is present much closer to the transcription start site, and this T series sequence located between -22 and -33 related to transcription start site serves as the binding site for the TRF1. So, the promoter selectivity by TRF1 is enhanced by some transcription activators as well as by its association with neuron specific TRF1 associated factors, and therefore, the same gene depending upon whether the TRF1 binds to this sequence or TBP binds to sequences can be differentially regulated. So, there is variations in the both cis acting elements as well as the general transcription factor binding to these sequences. Similarly, another TBP related protein called TRF2 was identified. This TRF2 again binds to TF2A and TF2B and actually displays very high conservation in the DNA binding domain. Remember, all these Tata binding protein family of genes, they are highly conserved in the DNA binding domain, but they do recognize variants of these Tata box sequences and have either specific tissue specific or cell type specific recognition, and their interaction with specific subset of tabs actually also are different. So, the TRF2 does not bind the Tata box containing DNA. Just like the TRF1 binds a different sequence called the TC box, the TRF2 also does not bind a Tata box containing DNA, and it fails to complement TF2D in base or activated in vitro transcription assays. Now, remember, it is again very important when you say TBP, when you purify for example TF2D from a specific cell type, and then if you replace the TBP present in the TF2D with a recombinant TBP, it will still act as a TF2D. But instead of TBP, if you now add TRF2 to the TF2D complex, it no longer substitutes the function of TBP, saying that TRF2 is actually a variant of TBP and it has a function that is different from the TBP. The reason why it cannot substitute TBP in the TF2D complex because TRF2 cannot bind to the Tata sequence. Now, the job of TF2D is to activate transcription from a Tata box containing promoters, but the TRF2 cannot bind Tata box and therefore cannot substitute for TF2D substitute for TBP in a TF2D complex. Instead, the TF2D actually associates with a DRA binding factor, which is another upstream activation factor, and it selectively regulates transcription from a DRA containing promoters. Now, remember my last class where I told you there are variations in co-promoter sequences. Some of the eukaryotic promoters instead of a Tata box actually contain what is called as a DRE motif. And here I am telling you there are protein factors which specifically bind to this DRE sequence, and a variance of a Tata binding protein is actually involved in this transcription activation from this DRE containing promoters. So, this TRF2 actually will recognize a DRE sequence instead of a Tata box. In fact, when people carried out genome wide analysis, there are now techniques to analyze what kind of protein factors can actually bind to what kind of sequences by techniques called chromatin immunoprecipitation and so on and so forth. When they carried out analysis to find out what kind of promoter sequences are actually occupied by TRF2 in Drosophila in vivo. When they actually do some of the studies, they found that several thousand promoters are actually occupied by TRF2 in vivo, clearly indicating that the TRF2 has a very important function in vivo. And very interestingly, many of these promoters which are occupied by TRF2 is not bound by TBP. That means there are subset of genes in vivo which are occupied by TBP, there are another subset of genes in vivo which are occupied by TRF2, clearly indicating that TBP and TRF2 may be regulating different sets of genes in Drosophila. And very interestingly, many of these promoters which are actually occupied by TRF2, they contain DREs, but they lack a canonical Tata box. So, very clearly that means the TRF2 is actually involved in the regulation of those eukaryotic promoters which are Tata-less promoters, but which actually contain DRE instead of a Tata box. So, you can see how variations in the Tata binding protein can actually contribute to differential gene regulation. There are genes which contain Tata boxes, and such genes are regulated by TF2D containing the normal TBP, and there are different sets of promoters which instead of Tata box contain a DRE, and such genes are regulated by this DREF and TRF2, which actually occupy such promoters and initiate transcriptionization. Very interestingly, when you knock out this TRF2, and in such mice they actually display defects in spermiogenesis and altered patterns of testis specific gene expression, clearly indicating that this variant of TBP, namely TRF2, is actually involved in the regulation of certain subset of genes that are essential in spermatogenesis or spermiogenesis. So, if you knock out this TRF2, the expression of these genes involved in spermiogenesis is blocked, and therefore you have defects in spermiogenesis. So, you have a tissue specific function for TRF2. There are other factors called TRF3, or which is also called as TBP2, which was actually discovered. This was discovered as recently as 2003, and the TRF3 was been extensively studied again in gonadal tissues as well as in early embryonic development. And in the case of mice, the TRF3 expression was shown to be increased very rapidly during oocyte growth from a primordial follicle formation until pre-ovulation, and at the same time the TBP expression is effectively eliminated. Very important. You see how differential gene regulation takes place. During the oocyte growth in the mice, it turns out during the primordial follicle formation, the expression of TRF3 actually goes up, and at the same time the expression of TBP comes down, indicating that the TBP function is taken over by TRF3 at this specific stage of oogenesis. And many recent studies actually indicate that this TRF3 actually has, as recently as 2007, detone TGen actually have shown that TRF3 has a very important role skeletal muscle differentiation. So, I am telling these examples to just to emphasize the fact that there are variants of TBP which seems to be having a very tissue specific and cell type specific or developmental specific regulation, okay, because their expression patterns is different than the normal TBP. And in the case, if you knock out this TRF3 in mice, the homozygous females display complete sterility due to oocyte arrest at the primordial follicle stage, which tells you clearly that the TRF3 has a very important role for further development from the primordial follicle stage onwards. And if you knock out, oocyte development cannot proceed beyond the primordial follicle stage, and therefore it leads to ovarian failure, clearly indicating that specific forms of TBP or variants of TBP have a very important role in oogenesis in mice. So, if you knock off this TF3, oocyte differentiation cannot proceed beyond a certain point. So, variants of TBP, TF2D subunits, whether it is TAFs or TBP variants, they play a very important role, and I have just given you some examples here. In the case of TRF1, in the case of Drosophila, it has been shown that TRF1 actually is essential for transcription of Pol3 as well. Certain promoters of Pol3, RNA polymerase 3 promoters are also regulated by TRF1, and in the case of TAF4B, it is essential for folliculogenesis in the ovary. In the case of mammals, there is a human variant of TAF9 called TAF9L, which is actually involved in apoptosis, and in the case of Drosophila, there are certain variants of TAFs which are expressed in testes, and these testis specific variants of TAFs are very important in spermatogenesis. So, what I told you so far is that variants of TAFs and tissue specific or cell type specific of expression of TAFs as well as TBP variants can contribute to differential gene regulation. That is the crux of what I told you so far, and it is now becoming very clear that TF2D variants have a key role in cell type specific development as well as differentiation. Alternate forms of co-transcription machinery components are used during metazoan developments of eukaryote to switch between transcription programs as cell differentiates. So, you can see in the case of eukaryotes, by simply expressing different forms of general transcription factors, you can bring about differential gene regulation. By simply expressing TRF1 or TRF2 or specific forms of TAFs, certain subset of genes can be activated or inhibited in different tissues and different cell types. So, differential gene expression can be brought about by differential expression of different general transcription factors, especially TAFs and TBP variants. So, incorporation of TAF4B in the place of one or two subunits of TAF4 allows TF2D to stimulate binding of certain DNA binding transcription factor to specific target genes, turning on a cell type specific transcription program. So, for example, in some of these gonadal tissues, by simply expressing a TAF4 variant, this TAF4 variant is now able to interact with specific subset of transcription factors, and therefore is able to drive a specific developmental program or a specific cell type specific fate in that particular time or space during development. Similarly, differentiation of skeletal muscle involves destruction of TF2D and replacement of a novel complex of TF2B containing TRF3 and TAF3, and this was discovered recently in 2007. What was shown very clearly in this particular paper by D2 and Tijan is that during skeletal muscle differentiation, the normal TF2D is destroyed and the normal TF2D function is taken over by a specific TBP called TRF3 and a specific called TF3, and they actually now go and activate subset of genes, resulting in differentiation of the skeletal muscle. So, you can see the variants of TAF and variant of TBP by selectively expressing in a specific time point during development and differentiation, specific genetic programs can be brought about, specific genes can be activated or repressed. Similarly, there are also examples wherein variants of TF2A can also contribute to differential gene regulation. Not only TF2D, not only TAFs or TBP variants, even variants of TF2A can contribute to differential gene regulation. For example, a cell type specific factor called TF2A alpha beta like factor called ALF has been identified in human testes as early as the late 2000, and this was shown to work in conjunction with TF2A gamma to stabilize TBP binding to the promoter. So, not only there are variants of TBP and variants of TAPs, there are also variants of other general transcription factors which can contribute to differential gene regulation, and this variant called ALF is also found in immature oocytes in frog Xenopus laevis, in which the ALF actually replaces TF2A during oogenesis. Now, what I told you so far is that at specific stages during development or at specific stages during differentiation in specific tissues, especially nervous tissue, gonadal tissues, etc., such specific variants of general transcription factors are expressed, and such variants of the general transcription factors in conjunction with certain upstream activators can activate say subset of genes contributing a specific differentiation program, like muscle differentiation, gonadal differentiation, nervous system expression of specific tissues during nervous system, and so on and so forth. So, I think what I told you so far in this particular class is that just as sigma factor switching in prokaryotes contributes to differential gene regulation by different sigma factors recognizing different set of -10 or -30 sequences in the case of prokaryotic promoters, general transcription factors can also be expressed differentially in eukaryotes, and this differential expression of general transcription factors can also contribute to differential gene regulation in eukaryotes. So, I have told you very briefly about how tissue specific or developmental space specific expression of certain variants of TAFs can contribute to differential gene regulation, and I also told you how variants of TBP itself like TRF1, TRF2, TRF3 can also result in activation of a different set of genes at different time points or in different tissues. So, an important point that you have to remember is that TF2D means it is not just there just to initiate transcript initiation from Pol2 promoters. There are variants of TF2D, there are variants of TF2A, and there are variants of other general transcription factors, and by selectively expressing these variants of TBP or TAFs at different time points during development and different cell type and different tissues, different set of genes can be activated or repressed, contributing to cell type or tissue specific regulation of gene expression. This is the crux of today's talk. So, as in the previous class, I have also given the number of original research articles here, which actually are the original research articles that have actually demonstrated how expression of different TAFs or different TBPs can actually contribute to different gene regulation in eukaryotes. And the other important aspect that we discussed today, and which is also mentioned in many of these papers, is that many of these TAFs actually contain enzyme modifying activities. They are actually enzymes. Many of the TAFs can actually either phosphorylate histones, they can acetylate histones, and this histone modifying activity of TAFs is playing a very important role in activation of transcription in the chromatin context. So, by their ability to modify histones, these TAFs can either make histones bind DNA more tightly or loosen histones from the chromatin, and this actually facilitates upstream activators to go and bind transcription factors, bind to specific elements, and pre-initiation complex can be assembled in the vicinity of these promoters, and this removal of histones is actually a very important component of transcription activation of eukaryotes. So, transcription regulation of eukaryotic promoters basically takes place by modifying histones, and the job of many of these TAFs is actually act as a bridge between general transcription factors and the upstream activator proteins, and the upstream activator proteins many times the ability by which they can activate or modulate enhanced gene transcription, they actually recruit these TAFs, and these TAFs in turn modify the histones or remove the histones in the vicinity of the Tata box or the transcription start site, so that the general transcription factors can come and bind to the Tata sequences and initiate transcription and enhance the rate of transcription. So, I think I will stop here, clearly telling you that the differential expression of general transcription factors or their ability to modulate or ability to bind to different sets of core promoter elements can also contribute to differential gene regulation. So, in the first three class I have basically covered various aspects of basal transcription of eukaryotic protein coding genes. How RNA polymerase II assembly takes place in protein coding genes, how in the first class I told you there is an RNA polymerase II which in conjunction with general transcription factors form the pre-initiation complex. Then in the second class I told you that there are variations in the core promoter sequences, there are variations of Tata box, there are many other core promoter sequences, and variations in the core promoter sequences itself can contribute to differential gene regulation. And the third class I told you, just like you have differential sigma factors regulating differential gene expression in prokaryotes, differential expression of general transcription factors like TAFs and TBP related proteins can actually contribute to differential gene expression, and therefore one important facet of eukaryotic gene regulation involves a general transcription factors around the core promoter sequences. In the next two classes we are going to talk about the role of general transcription factors in the activation of transcription by RNA polymerase I as well as RNA polymerase III. Because transcription initiation means not just protein coding genes, we also have to talk about transcription initiation from POL1 and POL2, how ribosomal RNA genes are transcribed, how tRNA genes are transcribed. So, we will discuss little bit about how tRNA gene pre-initiation complex is assembled on the tRNA gene promoters and how pre-initiation complexes are assembled on the rRNA promoters. We are going to talk about general transcription factors which are actually involved in this, and with this we more or less cover the basic aspects of transcription initiation in prokaryotes. And once we cover, then we are going to discuss that transcription initiation also plays the general transcription factors are not only involved in the transcription initiation, but they are also involved in transcription elongation, transcription termination, polyrelation, and so on and so forth. So, it is not just enough if the train starts off from a station, but it has to be monitored throughout and ultimately it has to reach a destination. Till then a number of processes are involved. Similarly, the general transcription factors are not only involved in transcription initiation, but they also contribute to transcription elongation, transcription termination, mRNA capping, polyrelation, and so on and so forth. So, with all, maybe in the next three classes we would have covered more or less all aspects of transcription initiation, and now the RNA, how RNA is actually synthesized from normal promoter. After this we are going to talk about upstream activation sequences, how and upstream transcription factors, how binding of these upstream transcription factors to specific sequences can bring about modulation of gene expression, and how these transcription factors can modulate the activity of general transcription factors binding to the core promoter elements, and what is the role of histones in all these things, and we are also going to talk about epigenetic regulation of gene expression. I think I will stop here.